Hello, welcome to the first of three presentations highlighting novel insights regarding the pathophysiology and genomic resilience of reproductive PRRS. I am John Harding, Professor of Swine Production Medicine at the Western College of Veterinary Medicine, University of Saskatchewan. In this first session, we will review the anatomy of the porcine maternal-fetal interface, or MFI. Before I go any further, I must acknowledge my collaborators who have spent countless hours contributing to our research on reproductive PRRS. We are a multidisciplinary team that has worked since 2011 in an attempt to solve some of the many mysteries related to reproductive PRRS. Our disciplines span clinical medicine, animal models, pathology, immunology, and genomics. This session has two specific objectives. First, to review the anatomy of the porcine placenta and maternal-fetal interface, and secondly, to understand the two basic mechanisms of nutritional support for fetuses. Because this is a prequel to two subsequent presentations, I would like you to think about these two important questions as they relate to PRRS transmission. Firstly, what are the important anatomical features of the porcine maternal-fetal interface? And secondly, how do those features potentially influence the transplacental transmission of large molecules and pathogens from the dam to the fetus? Now I should describe what I mean by the MFI because I will use the term repeatedly throughout this and my subsequent presentations. I will explain in more detail in a few minutes, but for now, it is sufficient to know that the MFI is comprised of the maternal uterine layers and fetal placental layers that are adherent or stuck together in such a way to provide nutritional support for fetuses and enable excretion of waste products. It might be helpful to think of the porcine MFI as two pieces of buttered bread that are stuck together, but with gentle force can be separated without tearing. This schematic shows a sagittal section of a fetus within a gravid uterus. The uterine layers include the perimetrium, myometrium, and endometrium. There are three layers of the placenta, amnion, allantois, and chorion, but in the pig placenta, the outer two layers fuse to form the chorioallantois or atlantochorion. It is the atlantochorion that is in direct contact with the endometrium, thereby forming the maternal-fetal interface. There are a couple of other interesting features to note in this image. Firstly, the amniotic cavity and fluids surrounding the fetus, and secondly, the allantoic cavity and fluids surrounding the amnion. Thirdly, there are avascular tips at the end of the atlantochorion which we think may be important for the interfetal transmission of PRRS virus because the avascular tips of adjacent fetuses overlap and are in direct contact with each other. This picture shows the placental units collected at gestation day 30 from a litter of 13. They are lined up on left and right, corresponding to the respective uterine horns, with the conceptae from the uterine tip at the top of the table and the conceptae positioned toward the uterine body at the base of the table. Each embryo has a discrete placenta, with the exception of identical twins, which occur rarely in pigs. The external surface of the placenta is the atlantochorion, and in most placenta, the day 30 embryo is clearly seen in the center. Note the variation in size and thickness of the placenta, which affects vascularization and ultimately the weight and potential viability of the fetus or the piglet at birth. There is no apparent relationship between placental size and the location within the horn. Pigs have a diffuse, non-invasive epithelial-chorial placenta. Diffuse because the MFI is continuous, unlike that of cattle, which have discrete areas of MFI known as cotyledons. And epithelial-chorial because the atlantochorion is in direct contact with a layer of maternal uterine epithelium. The epithelial chorial placenta is characterized by 6 cellular layers that separate the maternal and fetal blood, as listed in the schematic. There are 3 layers on the maternal side: the endothelium of the blood vessels, the endometrial stroma or lamina propria, and maternal uterine epithelium. Similarly, there are 3 layers on the fetal side: the fetal endothelium, placental stroma or mesenchyme, and fetal epithelium, referred to as the trophoblast. This is what the MFI looks like histologically. It is important to note that the placenta is a dynamic unit that remodels over the course of a normal gestation. This image shows the MFI at gestation day 85 and 105 on the left and right respectively. In spite of remodeling, the 6 layers remain constant. This image shows epithelium, stroma, and capillaries in both the maternal and fetal tissues. The 2 layers of epithelium are in very close apposition, so they appear as one layer at this magnification. Note how the placental stroma is much thinner at day 105 on right compared to day 85 on left. This is another H&E stained section of MFI collected from a gilt at gestation day 106. Note the tortuous, 3-dimensional orientation of the interdigiting maternal uterine epithelium and fetal trophoblast, referred to as maternal secondary folds. The maternal and fetal epithelium still appear as one layer at this magnification, but in this image one can clearly see sections of MFI that are linear, as well as cross-sections of folds. The placental stroma is light purple tissue on the upper aspects of the interdigitating epithelium. The endometrial stroma, or lamina propria, is filled with capillaries and uterine glands that supply nutrients to the fetus. If nutrients are supplied by blood, it is termed hematotrophic nutrition, but if nutrients are supplied through uterine glands, it is termed histotrophic nutrition. This is an electron micrograph that shows at high magnification the 6 layers separating the maternal and fetal blood. The gray areas at the top and bottom of the micrograph are the lumens of the placental and maternal capillaries at the top and bottom respectively. Working toward the center are the endothelial layers and a very thin stromal layer, which is represented as the light gray lines. In the center of the tissue is the interdigitating microvilli of 1 maternal and 1 fetal epithelial cell. Between adjacent epithelial cells in both the maternal and fetal sides of the MFI are very small interepithelial channels that facilitate the movement of small molecules from the maternal to the fetal blood. By late gestation, the distance separating the maternal and the fetal blood vessels can be as little as 2 microns. By contrast, the diameter of the human red blood cell is about 5 microns. The other type of fetal nutrition involves uterine milk, a whitish secretion containing proteins and amino acids produced by abundant uterine glands located in the endometrium. The uterine milk is secreted into placental areolae, specialized dome-shaped areas of placental epithelium which absorb the uterine milk by pinocytosis. This process is known as histotrophic nutrition. In this image, the small white spots are vesicles of uterine milk floating across the space between the uterine gland duct and the areolar epithelium. We think this may be a potential alternative mechanism used by PERS to cross the maternal-fetal interface, but this has yet to be proven. That brings this session to an end. I would like to thank our collaborators and funding agencies, particularly Genome Canada, Genome Prairie, and the Saskatchewan Ministry of Agriculture and Food. Additional details can be found in the following references. Thank you for listening.